Hi guys, welcome to the third part of my video, organization and expression of immunoglobulin genes. And in this part of the video we're going to learn how recombination actually takes place. Now let me just recall real quick what I talked in last video. So the way we create such a huge diversity in immunoglobulin genes is by having different variable regions. And for example, for heavy chain we have three different type of gene segments. One is called V, and we have a lot of these, and then we have a lot of another type of gene segment called D, and we have another type of gene segment called J, and we have a lot of these J too. And let's say this V can combine with this T, can combine with this J to make up one VDJ pair that would have a different antigenicity. It's gonna attack a different antigen. Then let's say if this V combines with this T combines with this J, this is gonna attack a different antigen, and this is the way it happens. So in this video we're going to learn the actual mechanism that how, let's say, this part of gene combines with this part of gene combines with this part of gene, and this recombination takes place in genetic level. It's not going to happen in mRNA, so it's not splicing, it happens in gene. The way this recombination happens is, let me just draw one V, one D and one J segment. So let's say I have a V, I have a D, and I have a J. And make a note that when I'm talking VDJ, I'm talking about heavy chain, because in light chain we don't have this D region, but everything else it remains the same. So let's say this is 5' part of the DNA, and this is 3' part, and we have another DNA strand down here, but we don't care about it because let's say this is the one we care about it. So what happens is, we have few sequence that I'm gonna draw by an arrow, and these genetic sequences are called RSS, or recombination signal sequences, or RSS. And these sequences detail a group of protein which are called recombinases. So these protein, they can identify the RSS, and they are gonna know where to cut. For example, we have a RNS right here and we have RNS right there. So this recombinase can identify that, oh, I'm going to cut over here, I'm going to cut over here, and these two are going to be joined. And there are different G proteins, but it's going to happen once. So one time it's going to be joined, and let's say D is going to be joined to J. And let's talk about how a recombination signal sequence actually looks like. So recombination signal sequence, it has a heptamer on one side. Heptamer means that there's 7 nucleotides, and in the middle we have 12 or 23 base pairs, and on the other side we have a nonamer. And this heptamer, it's palindromic. It means that it's going to be the same which way, doesn't matter if you read it. So let's say if we have a DNA strand, let's say a sequence, let's say this is our RSS, then we're going to have like 12 or 23 base pair. This is going to be our nonamer, this is going to be our heptamer. And if I read this way on like this part, and if I read this way on this part, I'm going to read the same. And if I have 12 base pair in my RSS, it's going to be called 1 turn RSS. And if I will have, let's say I have 23 base pairs right here, it's going to be called 2 turn RSS. So it can be either 1 turn or 2 turn. And let's say this V combines with this T. If this is 1 turn RSS, this has to be 2-turn. And let's say this part of this T combines with this, and let's say if this is 2-turn RSS, this has to be 1-turn RSS. Now if you guys got the pattern by now, 1-turn RSS always pairs up with 2-turn RSS. It's always like that. Let me just tell you guys few rules how RSS occur in different genes. So if we have a V lambda, or now I'm going to draw a lambda light chain. So let's say this is my V lambda, and in light chain I told you we don't have D, we just have V and J. And again there are a lot of V lambdas, but I'm just drawing one, and there are a lot of J lambdas, and I'm just drawing one. And the number, it differs in different species. Let's say if humans have 12, mouse can have 20 or 30. So if this is my 5 prime and this is my 3 prime, in lambda, this RSS that is attached to V lambda is always 2-turn. And I don't know if you need to know this, I don't know if your instructor wants you to know this, but I'm just telling. So if this is always 2-turn, the one that's attached to J lambda, it's always 1-turn. Let's say for kappa, this is V kappa, this is J kappa. Again, there are a lot of V kappas and a lot of J kappas, but I'm just drawing one. So the one that's attached to V is always 1-turn, and one that's attached to K, it's always 2-turn. And the rule of pairing of 2-turn RSS with 1-turn RSS, I think it's called 1-turn 2-turn recombination or something, but 1-turn always combines with 2-turn. And for a heavy chain, you have VDJ. I'll just use my picture over here. So the one with V, it always has 2-turn RSS. This one, the one on both sides of D, they are always 1-turn, and the one attached with J, it's always 2-turn. So 2-turn combines with 1-turn, this goes away, and we have VD and VDJ. So this whole section, they are removed when recombination happens, and we get a gene with V, D, and J. 1V, 1D, and 1J. And like I told you before, like out of all the Ds that are present, some of them could be pseudogenes, or genes that don't code any protein at all. So this VDJ recombination might be useless at some time if the VD or J picked up is a pseudogene. It can happen. And the enzymes I called that make this happen are called recombinases. And there are two different recombinases enzyme called RAG1 and RAG2. And the name RAG1 and RAG2, they stand for recombination activating gene 1 and 2. Now I told you this is an enzyme, this is a protein. So this is the gene, recombination activating gene 1 makes up this RAG1 protein, and recombination activating gene 2 that makes up this RAG2 protein. So these are the enzymes that make this happen. Now we're going to look how the mechanism of how this actually happens. So at first, let's say we have a V kappa gene. I'm actually going to be using a new paper. Now, let's say I have this V kappa, and let's say I have this J kappa. And the first thing that happens when you have to like remove these two is first they orient, and for example they orient something like this. of this. And they do this just to make it easier for the enzyme to cut them off, and it's actually the recombinase enzyme that identified this and make it arrange like this. And if we have, let's say this is like one configuration, and if we have this, let's say this VK, and this arrow points in this direction, and then we have this JK, and by these like arrows, these are my RSS, and these blocks, they are my genes. So if this is like this, they are going to arrange like this, and this is my first step. So you can see how this one in goes and attaches over here, and this is how it is. Because my video is 10 minutes already, I'm going to make another video